Okay. So Bismillah ar-Rahman ar-Rahim. So today's lecture is on nerve transmission, okay? So which is about the conduction or the propagation of action potential down the axon. So we will learn about the basic structure of neurons, and we'll talk about conduction of the action potential or propagation of the action potential in the axon, as well as the factors that affect the speed of the conduction of the action potential. Okay. So as you guys are familiar with this is a neuron. This is the cell body of the neuron. It's called soma as well. So these branches here are the dendrites, okay, from which the neuron will receive the signal. This is the axon, and this is the axon terminal. Now, from previous lecture, we said graded potential is generated at which location? Yeah? In the dendrites. Very good. So, as you can see here in the dendrites, the release of the neurotransmitters would open the ligand-gated ion channels and would allow ions to move in the dendrites. This will make either depolarization or hyperpolarization of the membrane, depending on which ion will be moving to the inside. When the graded potential reaches this location here, which is the axon hillock, if it brings it to the threshold, if it will make it depolarize, making it, reaching the threshold, this will open the voltage-gated sodium channels, and an action potential will be generated that will be propagating down the axon. It will just move down the axon to reach the axon terminal, okay? So what's special about the axon hillock, as we said, is there is the huge, amount or expression of the voltage-gated sodium channels. So in the other structures like the dendrites and cell body, we don't have much of the voltage-gated channels, okay? They are more concentrated in the axon hillock as well as in the axon. As you can see here, in red is where there is high enrichment of the voltage-gated sodium channels, okay? And so as we learned that at the axon hillock, if the stimulus or the graded potential was high enough to bring the membrane in the axon hillock to threshold. So as you can see here, the threshold in this case is shown here. So if it brings it to the threshold, this will open the voltage-gated sodium channels. And when the voltage-gated sodium channels are open, the sodium will move to the inside of the cell, right? Because the equilibrium potential for sodium is around positive 60 millivolt. Okay, so it's around that. Okay, there is a range, but it's around positive 60 millivolt. And so the membrane at this stage is in the very negative values, like around negative, 75 as shown in here. Okay. So different neurons, they do different values, but there is a range, okay? Now, when the membrane reached the threshold, the voltage-gated sodium channels would open, and sodium will move to the inside to bring the membrane closer to its equilibrium potential. But it doesn't reach the equilibrium potential for sodium, right? Why is that? Why it stops at this level here around positive 30 millivolt? Why is that? Yeah? Because of the inactivation gate, right? The inactivation gate of the sodium voltage-gated channels would be closing, and we have the opening of the voltage-gated potassium channels. When the voltage-gated potassium channels are open, we know that the equilibrium potential for potassium is around negative 94, okay, something like that. And so it's in the interest of potassium to move outside of the cell in order to take the membrane closer to its equilibrium potential, okay? And as you can see here, it even goes further than the resting membrane potential because the potassium voltage-gated channels are slow at closing. Once they are closed, the action of the sodium-potassium pump will restabilize the resting membrane to its level or to its status. Okay, is this all clear? Okay, and so what happens is that once we have a segment of the axon, so let's say here we have the initial part of the axon, and here is towards the axon terminal, the synaptic terminal in here. Okay, so what happens is that once we activate the voltage-gated sodium channels in this part, in point A, sodium will move to the inside of the axon, okay, and will depolarize this part, okay? Depolarization of this part will bring the next part, which is in this case here, will bring it to threshold again. Sorry, so I'm just doing, so let me just take that, and okay. So these are the sodium voltage-gated channels. So once they are depolarized, okay, once they are open, sorry, this will depolarize the membrane as sodium moves to the inside, okay? And as this part of the axon is depolarized, this will affect the next or the subsequent voltage-gated sodium channels, causing depolarization of that part, the next part, and this will open the voltage-gated sodium channels, and they will enter to depolarize the next part, okay? In the same time, we have the potassium voltage-gated channels open to repolarize the membrane. So it's like a cycle. We have the voltage-gated sodium channels are being open, okay? Entry of sodium depolarizing this part A of the axon. This will affect the voltage-gated sodium channels in part B, okay? And in part A, we have the voltage-gated sodium channels being inactivating, and opening of the voltage-gated potassium channels to repolarize the membrane. So it's like a fuse, okay? Once you have one part being, burning, it will burn the whole thing, okay? So once we have point A being depolarized, the voltage-gated will just affect, the next part, etc., until the action potential will be traveling down the axon terminal. Is this all clear? Okay. And so there are multiple factors that affect the speed of the action potential, okay? Namely, there are two factors. There is the diameter of the axon. Remember, like, the water hose, if it's large, okay, this means that we will have a quicker and a huge amount of water moving in, right? Whereas if the hose was smaller, the diameter, it means that the flow will be slower, okay? Now similarly with axons, we have multiple types in our body, okay? So we have, for example, the A alpha type, which is, which has a huge or a large diameter, and this, the conduction or the speed of the action potential tend to be, faster. As you can see here, it has a conduction velocity of 80 or 120 meters per second, okay? Whereas the C type has a very small diameter, as you can see here, this is in micrometer, and thus the speed is slower, okay? There is also another factor, which is myelination. As you know, probably you're already exposed to this. Myelin is basically, consists of lipids that cover up the axons, as we will see in a second. And myelination tend to speed up the, the action potential. And so the type of action potential conduction in myelinated neurons is called salutary conduction. Okay, and as you can see here, the A alpha is myelinated, whereas the C type of axons is unmyelinated. Okay, so there is the diameter as well as the myelination of the nerves that determine, how fast the action potential will be traveling. So again, this is about the diameter. We have the A alpha type, which we said has a large diameter. And we have the C fibers, which also, have very small diameter, okay? So as you can see here, the A alpha is connected to a certain receptors, like the proprioceptors. Anyone is familiar with the proprioceptors? Okay, so the proprioceptors are the receptors that are found in our skin, okay, or near joints, which inform our brain of where our limbs are, okay? So this type of information has to reach the brain very fast, okay? So proprioception reaches the brain very fast because it's conducted, the signal is being transmitted in the large diameter axons, the A alpha type. Whereas the C type of fiber is linked to the nociceptors. These type of receptors are the ones that respond to noxious stimulus, okay? When you cut yourself, or when you injure yourself, or you have inflammation, these are the nociceptors that innervate our skin, that respond to the noxious stimulus, okay, and will transmit the signal to the brain, and you will feel the pain. So pain doesn't need to be traveling very fast, okay? So it travels in the C unmyelinated with the small diameter axon, okay? Now with myelination, myelin in the central nervous system is produced by which type of cells? Which glial cells? In the central nervous system? Oligodendrocytes, right? And in the peripheral nervous system? The Schwann cells, very good. So these cells will wrap around the axon, they will wrap around the axon, and so they will insulate the axon. So they, myelin has, some types of lipids in there which make up a very nice insulator. It's like, it will concentrate the voltage-gated channels in these gaps here. As you can see here, we have the myelin here, the myelin sheaths. And the gaps in here where we have the concentration or the expression of the voltage-gated channels, okay? And so what happens is that the action potential will jump, okay? Because these myelin sheaths, they are very good insulator. They don't allow the escape of the ions, okay? They insulate the axons in order to minimize the loss or leakiness of the ions. And so the action potential will be traveling very fast by jumping from, one node of Franvier. So this is, the gap here is called node of Franvier, where the action potential will be jumping from one node to the other where the voltage-gated channels are, okay? And so you can see here in an areas of the neurons where we don't have, voltage-gated channels, or we don't have myelin, the signal or the graded potential tend to decay, as we said, right? Whereas in the, you can see here, unmyelinated axon, this is unmyelinated axon, we don't have myelin, okay, but we do have voltage-gated sodium channels and potassium channels. And so the action potential will propagate, but it's going to be slower than the myelinated axon. So this here, you see that the axon is myelinated. And so we tend to, there is the concentration of the, voltage-gated channels in the node of Franvier, and myelin will insulate the axon, and so the action potential will jump from one node to the next until it reaches the axon terminal. So in the myelinated axons, we have a higher speed of action potential than the unmyelinated axon. And so in certain diseases like multiple sclerosis, multiple sclerosis is an autoimmune disease where in the patients there is the production of antibodies, okay, which are abnormal to attack myelin, okay? And so there is the loss of myelin, and not just myelin that gets affected, even the axons get affected, and so this will affect the propagation of the action potential, okay? And so depending which nerves are being affected in the patients, thus the clinical manifestations. So some In some patients, there is the loss of myelin in the optic nerve, and this will affect, their vision, okay? So it depends where the effect is, okay? It's very devastating disease. Now the question is, once the action potential, so we said it's traveling in the axon, once it reaches the axon terminal here, what does it do? What will happen is that when the axon reaches the axon terminal, as you can see here, what happens is that the depolarization of the axon terminal will activate a voltage-gated calcium channel, okay? And so now calcium is gonna move from the outside to the inside. If you remember from previous lectures, we said calcium is very important second messenger, right? Plays very important role in the cell. And so the entry of calcium will trigger the release of these synaptic vesicles. So these here are called synaptic vesicles. If you remember from early lectures, we said that synaptic vesicles are derived from which organelle? Do you remember? Synaptic vesicles, or vesicles generally, transporting vesicles, from the Golgi apparatus, right? They are made, from the ribosomes, then they move to the, endoplasmic reticulum, and then to the Golgi, and then they get transported by the help of which type of cytoskeleton proteins? Microtubules, very good. So they will get transported by the motor proteins to the synaptic terminal, and here they are stored, waiting for the synaptic terminal to be depolarized, and the entry of calcium will trigger the fusion of these synaptic vesicles and the release of the neurotransmitters in the synaptic cleft. So this is called synaptic cleft, okay? So this similarly, so here once the axon terminal is depolarized, we have the activation of the voltage-gated calcium channels. When the calcium moves to the inside, it will trigger the fusion of the synaptic vesicle and the release of the neurotransmitters in the synaptic cleft. Now these neurotransmitters are ligands, so they will bind to the postsynaptic receptors here. You see those on the next neuron, in the dendrite of the next neuron. There is the expression of the ligand-gated ion channels. So these, when we have the neurotransmitters are bound to the ligand-gated ion channels, they will open. And so when they are open, the ions, depending on which ions these ligand-gated ion channels are specific for, they will either hyperpolarize or depolarize the next neuron, okay? So for example, the movement of chloride from outside to inside will make the cell, the next neuron, hyperpolarized, isn't it? And the movement of sodium to the inside will make it depolarized, right? Okay. Now, not just, this is how, neurons communicate with neurons. Also, this is how neurons or nerves communicate with other, other cell types like skeletal muscles. So, as we said, we have this neuron here. This is the presynaptic neuron, and we have here a synaptic cleft, and this is a muscle cell, a skeletal muscle cell. And so when this neuron is depolarized, this will activate which voltage-gated channel? Calcium, right? And calcium will move to the inside and will trigger the release of the synaptic vesicles, the neurotransmitters. In this case, it's acetylcholine. Acetylcholine will bind to the cholinergic receptor, which is a ligand-gated ion channel on the muscle cell, okay? And when the ligand-gated ion channel is open, the potassium will move, or sodium will move to the inside, okay? And this will depolarize the muscle cell, okay? And once the muscle cell or the skeletal muscle cell is depolarized, this will activate again an L-type or voltage-gated calcium channel. And so calcium will move to the inside, and this will trigger what's called calcium-induced calcium release. When there is a high concentration of calcium in the skeletal muscle, this will open the ryanodin receptor. This is called ryanodin receptor, which is here in the sarcoplasmic reticulum in the muscle cell, and will allow the outflow of calcium. And so when there is a huge amount of calcium in the skeletal muscle cell, this will trigger the contraction of the muscle cell. Okay, so we don't need to go in much details because you're going to learn about this in the next block. Okay, but this is how a neuron communicates with a muscle skeletal cell. Okay, so when the neuron is depolarized, they release, the synaptic neurotransmitters, the vesicle neurotransmitters. They will communicate with the muscle cell to depolarize the muscle cell, and this will trigger the release of calcium in the inside, which will induce the contraction of the skeletal muscle cell. Okay, is this all clear? Okay, now here is some clinical applications that, in medicine where they take, advantage of the voltage-gated sodium channels, like the local anesthetics. When you go to the dentist, they give you a local anesthetic to reduce your pain. And so these local anesthetics, they work by blocking the voltage-gated sodium channels, okay? So we said that the type of nerves that conduct the signal in response to a noxious stimulus are the nociceptors, right? These are the nerves that will convey inform your pain of the noxious stimulus and give you the feeling of pain, okay? So these nociceptors, what they do is they basically, in medicine, for example, when you go to the dentist, they give you local anesthetics. And so these local anesthetics will block the voltage-gated sodium channels. And so in this way, the pain signal or the response of the nociceptor would be stopped. And so this is how they elevate pain, okay? So this is the end of the lecture. Any questions? Okay. Any questions? Yeah? Which one, the nociceptors? Very good question. It's not just the nociceptors that are active in the reflex arc. There is also the other sensory neurons, like the ones that are responsible for touch. Okay, these are myelinated as well. Okay, so there are multiple sensory, type of sensory receptors, including the nociceptors. No, there is more details I can tell you after this. Okay. Any other questions? Yep. I can't hear you. So let's take attendance first. Let's take attendance. So guys, don't forget to take the test, the self-assessment test. Assess yourself, okay? And tomorrow, I think we have a review session, right? At 8, we will go over some of the, complicated questions, okay? Yeah, this is the LGD, it's just a review, okay?